All right. So today's focus is actually on one of, many of you know us for the AMP-CMD-KP, before that, the Type 2 Diabetes Knowledge Portal, but we were very lucky, from NIH really, was very supportive in expanding our AMP-CMD resource to a community genomic resource for all of common disease. And so this resource has been available for about a year and a half now, and it has 15 community portals within it, but really is meant to be a place for taking from association via GWAS to function. And how do we do that, right? So that's a process, and it's an incredibly important biological model. But today we're going to hear about a piece of that helps to build on this effort to really take from GWAS locus to a mechanism or a gene or a pathway of interest. So with that, that whole resource rests on a biological model that assumes that causal variants lead to, or organismal function. And the challenge with that is that that's what GWAS does for us. It gives us regions and tons of them, right? We all know this. But the space in between is the challenge. And there's, what is the regulatory effect that causal variant may have? In what gene? What are the cellular functions? In what tissue? All of the work here is really important to bring to bear to actually go from those GWAS loci to an organismal function. And so, for the A2F knowledge portal that we've built as an expansion of the CMDKP, we aim to integrate data and represent knowledge from that space between and all the GWAS results that have predicted these associations. And this is an ongoing effort, right? So some of the data types are still nascent. Some of the models and the approaches are really well developed, but haven't been run systematically, and so on and so forth. But our approach is really to try and capture that space between through summary representations organized by scientific questions to represent knowledge and results on the A2FKP. However, right, so with that, here's what we enable, right? Right now, if you are a person who's trying to, determine what gene you might want to study, you have to do historically, you used to have to do this long, arduous task of going to different places, right? But with the knowledge portal, really we aim to simplify that and put it into one place so that you can see all the GWAS loci for your favorite gene or trait, disease or trait, and see all the genes that are associated with that and all the relative functional annotations and bioinformatic methods that are incorporated therein. So we try to make this easy, but our approach in doing that is specifically in collaborating directly with the researchers and the investigators. And that's what's critical here in the sense that, we are a resource that presents data and knowledge, but we do this by collaborating with the community, not only for the data that we present, but the methods and the approaches. And that's really key because that's really what's pivotal to today's conversation. So in this space, right, you could have, large efforts like GTEx, and you can have large efforts like the ENCODE and large efforts like what CMDGA is doing to work with us to bring all the genomic annotations together. But certain people have really great work in certain areas. Particularly today, we're going to focus on QTLs and colocalization. And while we have ongoing efforts in this space, right, we have a systematic effort to, lead a mission that Michigan is leading to build a resource called Colocus to visualize and query colocalization from common diseases. And that's available in a very early version on the portal right now. You can go to the portal on the KP Labs tab and visualize our Colocus module with one data set, right? And that's great. We also can store and process EQTLs at scale. But in the background, we need to make this accessible. So a postdoc that works with Jason, Miad Zindavi, is implementing SUSY, which is critical for running fine mapping across all of our traits. And those will be implemented for the spring, but then you got to add to that a colocalization approach. And so we're implementing Colocus on top of it with results for the summer. So this is our systematic approach. And that will take time, right? So this will, again, produce colocalization results on close to 1,000 traits in the portal and make those accessible and visualizable through Colocus. However, that work takes a long time. But when you have a great collaborator who you happen to know for a long time and work's already been done in this area, particularly in a disease that this group is an expert in, and they want to make those data sets and results available to a wider community, that's where you talk about working with Ayellet to build a microportal. So you're going to hear about this work today. And the reason we enable this is, these things can happen faster, and getting these results out, not just in a publication, but in a visualization that you can access. We have built a sort of a micro portal suite to enable these results to fast track to the to the user community, but without being fully integrated into the portal yet. That'll come later, right? So this is what we call Be Your Science, or bring your own results. And this is, something we've been doing for several years now to enable sort of vignettes of data getting to the wider resource community without being fully integrated into the portal, because they're done by the community, how they want to visualize the results, and they can be fast-tracked because they can build these resources themselves with a little help from us, or they can partner with us to build the larger. So that's what's going on here with Ayellet, and she's going to talk about that work. But you can do this yourself if you want to build these resources, and they're all freely accessible, and we really love to work with you to build them, to customize them to the features and the results you'd like to see on the site. So with that, you're going to hear about vision genomics today, but you're also going to hear about actually the most important part, which is the scientific research that went into it and the results that Ayellet's team so graciously has collaborated with us to represent. So with that, I'm going to turn it over to Ayellet, and she's going to walk you through both her results and the portal itself.
Thank you, Noelle. That was an excellent introduction to my presentation, and I'm really excited that we get to work together again after many years.
Me too.
So I'll share my screen. Okay, so you see my screen okay?
Yes, thank you.
Okay, great. So in my lab at Mass Eye and Ear, we're interested in integrating GWAS with functional genomic data and prior biological knowledge to shed light on the causal mechanisms, genes, pathways, pathogenic cell types that lead to complex diseases, with a focus over the past years on common retina-related diseases. And a lot of the work that we have done over the past few years have been on glaucoma, and I'm going to tell you today about some work in that realm. So the outline of my talk, I'm going to give you a brief introduction on primary open-angle glaucoma. That's the most common form of glaucoma. Tell you about what's has been discovered so far with respect to GWAS for this disease. And then I'll discuss some work that we have done to try and prioritize the underlying causal mechanisms and genes using colocalization analysis of expression and splicing QTLs and Mendelian randomization of co-occurring QTLs and GWAS loci. I'll briefly touch upon some work we've done to try and predict the prominent cell types that may lead to glaucoma, looking at enrichment of these QTL map genes to GWAS loci and single cell eye expression data. And finally, I'll show you how we began to integrate both the results from the colocalization analysis and Mendelian randomization into the Vision Genomics portal, and our kind of pilot of starting to inspect glaucoma GWAS loci, and eventually we'd like to add other ocular traits to this portal in relation to EQTLs and other functional genomic data. So primary open-angle glaucoma is a leading cause of irreversible blindness worldwide. It affects over 70 million people. And it's characterized by gradual cell death of the retina ganglion cells that are in the posterior part of the eye. So if you have here the lens and the cornea, and the retina is a thin layer, complex layer of neuronal and non-neuronal cells along the posterior part of the eye. The retina ganglion cells are the cells that translate the light vision signals to nerve impulses and transmit the nerve impulses to the visual cortex to display the images we see. And so POAG, primary open-angle glaucoma, is characterized by progressive degeneration of the optic nerve, which is really the axons of the retina ganglion cells, which eventually can lead to visual field loss. A major risk factor is elevated intraocular pressure. And one of the few structures that are thought to have been shown to affect and cause elevated eye pressure, it's kind of hard to see on the cartoon, but it's what we call the outflow pathway. They're both a conventional and unconventional outflow pathway in the front of the eye. The conventional outflow pathway is made up of a trabecular meshwork through which lymphatic vessels called Schlemm's canal go through, and the aqueous humor, the fluid that flows through the front of the eye to clear the eye of debris and microbes, flow through this lymphatic vessels, and inadequate drainage of this fluid is thought to lead to potential blockage and elevated eye pressure. There's also this non-conventional outflow pathway, the uveoscleral, that's a ciliary muscle, and the aqueous humor flows through this tissue as well. And its inadequate drainage is also thought to affect intraocular pressure. But in addition to this major risk factor, about 40% of POAG patients have eye pressure in the normal range, what we call normal tension glaucoma. And those are the kind of trickier patients to treat, since to date there is no cure for reducing the sensitivity of the retina ganglion cells. The best treatment to date is lowering the IOP levels. So some of the kind of open-ended questions are, what are the genes, biological processes, and cells that are affecting intraocular pressure, homeostasis, and POAG risk in the anterior segments of the eye, in particular in the outflow pathways? And what are the genes and processes and cells that are affecting the retina ganglion cells in the back of the eye? Is it dysfunction in the retina ganglion cells themselves, or other cells that support the RGC, such as neuronal support cells or macroglial cells? So to date, a very large cross-ancestry meta-analysis has been conducted on POAG, published in 2021, over 34,000 cases and about 350,000 controls, comprising of European, African-American, and East Asian individuals. And that led to the identification of 127 loci, explaining about 9% of the heritability. And also a subset of European samples, GWAS on the European subset has been conducted on 16,000 cases and 200,000 controls, leading to 68 loci, over 20 of which were not found in the cross-ancestry meta-analysis. And the largest to date meta-analysis for IOP was conducted across 140,000 individuals, primarily from the UK biobank, so primarily of European ancestry, leading to the detection of 112 loci, explaining about 9 to 15% of its heritability. And more recently, there's a large multi-trait meta-analysis that was published from Stuart McGregor's lab that constituted both POAG cases as well as IOP and vertical cup to disc ratio, which is associated with POAG risk, and that led to the detection of 300 loci. So in the future, we're excited to also look at that larger meta-analysis. And there's ongoing efforts led by Anthony Khawaja and Piero Heisi also to increase the IOP meta-analysis and perform a very large cross-ancestry meta-analysis. So there are larger and more powerful GWAS in the pipeline for these traits. So the big kind of million-dollar question is, how do we go from these associated common variants with complex traits to the underlying causal genes and pathways and causal cell types through which they exert their effect on the complex disease? And there are several challenges to doing so. The first is that due to linkage disequilibrium, there are often multiple genes that are tagged by the associated variant. There's often allelic heterogeneity, more than one causal variant tagged by the associated variant. The majority of GWAS variants for POAG, as with other complex diseases and traits, lie in non-coding regions, making it hard to pinpoint the affected gene or genes just from the associated variant itself. And given that the majority lie in non-coding regions, and today we know from many studies that they have regulatory effects on complex diseases, we have seen that the regulatory effects both may affect distal genes and may vary by tissue and cell type. So ideally, we'd like to measure their effects of the regulatory effects in the relevant tissue and cell type. So the underlying working hypothesis of our work is that most of the associated variants with POAG and IOP will affect the disease, the trait, through modulation of gene expression levels, alternative splicing, and the affected genes will cluster in specific pathways, and the expression will cluster in specific cell types and tissues that are important for the development of the disease. So just a very brief introduction. I'm sure many of you are familiar with this. So how do we detect these genetic regulatory effects? Well, expression and splicing quantitative trait loci is one approach. where we collect and perform RNA sequencing on multiple samples from multiple donors for a given tissue, ideally more than, for enough statistical power, more than 70 samples per tissue. And we test genotype data within a cis window, within 1 megabase around the transcription start site of each gene that is expressed in the tissue using either imputed genotype data or whole genome sequence from the donor's DNA. And using linear regression analysis, we identify variants whose genotype is associated with expression or splice events, correcting for various covariates, including hidden inferred covariants on the gene expression or splicing, genotype PCs to correct for any population stratification, sex, and other covariates that might be important for the specific study. For the splice QTLs I'll show you today in GTEx, and we have an ongoing project in retina with Anand Swaroop's lab. We detect splice events. We have detected splice events using leaf cutter, which can detect different types of splice events, such as exon skipping, alternative starter-end site, 5' or 3' starter-end sites of exons, more complex splicing events. And the way that leaf cutter measures, quantifies a splice event is the ratio of number of split reads that map to a given start-end splice event divided by the total number of split reads within an intron excision cluster, such as in this exonic skipping example. So using that splice quantification and appropriate gene expression normalization, EQTLs and SQTLs have been computed for about 49 human tissues in the GTEx project, based on 838 donors with whole genome sequencing data, anywhere between 70 to 700 RNA-seq samples per tissue, and from Anand Swaroop's lab in peripheral retina, based on 400 RNA-seq samples. And you can see that the discovery of EQTLs and SQTLs is proportional to the sample size to power of detection. And EQTLs have been discovered to date for almost all protein coding genes and about 80% of the link RNAs, and SQTLs for about 80% of protein coding genes and about half the link RNAs. And I'm just to show you the tissue specificity distribution of the EQTLs and SQTLs, and why we believe as a starting point, before we have EQTLs and SQTLs in all the relevant glaucoma tissues, why the EQTLs and SQTLs and GTEx may be relevant for starting to investigate the regulatory effects on glaucoma. If you look at this tissue specificity distribution, it's bimodal. So the majority of E and SQTLs are shared across multiple, or the majority of the GTEx tissues, well, about 20% of the EQTLs or 30% of the SQTLs are more tissue specific. And this is likely due to sharing of cell types across tissues, or to genetic regulation that's just shared across cell types and tissues. And in GTEx, there are various cell types that are relevant for glaucoma, such as the arterial tissues, brain tissues that have neuronal and neuronal support cells, and fibroblasts, as I'll show you later. So first we asked, how relevant these EQTLs and SQTLs may be for glaucoma. And so we tested whether any of the EQTLs, SQTLs from the 49 GTEx tissues were enriched for glaucoma associations. And using a method we developed called QTL Enrich, it's available on GitHub, we found significant enrichment of glaucoma associations and IOP associations amongst most of the GTEx, EQTLs, and SQTLs. Shown here are the tissues that show the most significant enrichment, including fibroblasts, lymphocytes, artery, and brain tissues for EQTLs, or artery and brain, and arterial heart appendage for the SQTLs. And this analysis that corrects for potential confounders, such as distance to TSS and MAF and local LD, suggests that several hundred, over 400 EQTLs may be new associations with glaucoma, and over 200 SQTLs may be new associations with glaucoma. So we next asked whether focusing on the known GWAS loci, the strongest associations, whether the EQTLs and SQTLs in GTEx and the retina EQTLs can help prioritize causal genes in the 127 POAG variants loci from the cross-ancestry meta-analysis, the 68 from the European meta-analysis, and 133 IOP variants in 112 loci for IOP. And for this, we applied two colocalization methods, the Bayesian-based approaches, eCaviar and NLOC, that are based on taking in as input the summary statistics from the GWAS and the EQTL, and perform fine mapping, take into consideration the local LD and the allelic heterogeneity. They assume that there could be more than one causal variant in the region. And this is just a toy example. The figure was adapted from Bartolome et al., the Coloc, the original Coloc method. And this is just an example where two co-occurring GWAS and QTL signal are not tagging the same causal variant. This is a locus compare showing the minus log p-value of the GWAS versus that of the EQTL. And this is an example where they are tagging the same causal variant, suggesting that the QTL may be the underlying causal mechanism for the GWAS, for the complex trait. And so these colocalization methods, they output a posterior probability that the GWAS and QTL signals are tagging the same causal variant. And so we applied this to all of the POAG and IOP loci, and found significant colocalization for about 60% of the POAG and IOP loci, suggesting over 200 genes in 76 POAG loci and about 280 genes for 79 IOP loci. Some of them were overlapping, such as the TMC01 locus or GAS7, and some seem to be POAG specific, independent of IOP, such as the famous CDKN2AB locus that's associated, the strongest association with normal tension glaucoma and a glucose transporter. For about 20% of the loci, we got significant colocalization with both methods. So these are prioritized as the highest confidence results, but casting a broader net with two methods, given that they're slightly different methodologies and underlying assumptions, we were able to prioritize genes for 60% of the loci. 10 of the loci colocalized with an retina EQTL. And on average, we were able to reduce the number of genes that map to a given locus just based on having EQTLs in the locus from an average of about 23 ES genes tested per locus to an average of 3-4 different genes prioritized per locus. So I think one interesting area of research in the future is to understand whether this is because we're not yet focusing just on the tissue of action for the disease, or whether many of the common variant associations in the future will turn out to be affecting more than one gene that has an effect on the complex disease per locus. A single causal gene was proposed for about a quarter of the loci. And while the majority of the genes, 66% are protein coding, 20% are non-coding, and the rest are other biotypes. And for 30% of the colocalizing genes, they are not the nearest gene to the lead GWAS variant. We've seen this with other complex diseases and traits, demonstrating the importance of measuring regulatory effects of the variants. So we next asked whether the target genes of the ENS QTLs that colocalized with the POAG and IOP GWAS loci were enriched in specific biological processes. So we tested enrichment in both gene ontology, reactome, and KEGG. And we found that the strongest significant enrichment, FDR below 0.01, of the glaucoma genes in elastic fiber formation and extracellular matrix organization. This has been previously shown. And for the IOP genes, we saw significant enrichment in transcriptional regulation by VENT-X. It's a transcription factor, a homeobox factor that affects embryonic development, including neural crest development and hematopoiesis. And we found that it's expressed in immune cells, both in the front and back of the eye, suggesting a potential role of the immune system in IOP regulation. And then the IOP genes were also enriched in vasculature development, as well as in more basic cellular processes, such as regulation of cytoskeleton organization and cell adhesion. So to show you an example, kind of zoom in on a given gene, a given SQTL, we'll look at EFMP1, which is an EGF-containing fibrillin extracellular matrix protein. And this is a locus zoom plot showing the GWAS p-value for POAG from the cross-ancestry meta-analysis. The colors color-coded based on their LD to the lead GWAS variant. And you can see that EFMP1 falls outside the main highest LD region around the lead GWAS variant. But it turns out that this signal significantly colocalizes at a posterior probability of 0.51 with an SQTL acting on EFMP1. And this SQTL leads to skipping of exon 6 and 7 in this gene. And the colocalization analysis suggests that skipping of these two exons may be protective for POAG. And so I'm also showing here kind of the gene model, and the splicing events color-coded based on the splice event that is associated with that was significantly colocalized and that's associated with POAG risk. And these are features that we'd love to add to the Vision Genomics portal to be able to more interactively investigate the underlying splice events that are potentially associated with disease risk or trait variation. Interestingly, separate study of looking at whole exome sequencing in Filipinos families led by Ryan Colantis and Janie Wiggs from Mass Eye, they found enrichment of rare mutations in the EFMP1 in families with juvenile onset open-angle glaucoma. So supporting, this gene is likely the causal gene. The colocalization analysis and looking at EQTLs in the context of GWAS associations can also propose direction of effect. So this is another example, GAS7, it's a growth arrest specific gene. An EQTL in fibroblasts is significantly colocalizes both with POAG risk and with IOP. And looking at the direction of effect, decrease of expression is proposed to decrease IOP levels and POAG risk. Another example that seems to be IOP independent is EQTL acting on SLC2A12. It's a transporter that catalyzes the uptake of sugars through facilitated diffusion. And retina EQTL, in addition to other GTEx EQTLs, significantly colocalized with POAG. This locus is not associated with IOP. And looking at epigenetic data from Anand Swaroop's lab from NEI and Jayshree Advani generated this figured analysis, we see that the lead GWAS variant and the EQTL with this transporter overlap cis-regulatory element inferred from epigenetic data, providing epigenetic data from human retina, so providing additional support that this may be the causal mechanism. And finally, some of these looking at the various independent signals in loci, we find that some of the EQTLs are colocalizing not with the primary GWAS variant, but with a secondary signal. For example, this angiopoietin-1 brain EQTL from GTEx that colocalized based on NLOC at a posterior probability of 0.23. And here, increase of expression of angiopoietin-1 is associated with decrease of IOP and protection for POAG. And angiopoietin-1 has been implicated in vascular development, and mouse models are knockout of this gene in mouse models are in line with this direction of effect. So we're kind of interested also to add this information about whether the colocalization is with the primary, secondary, or tertiary GWAS signals. Just one word of caution, and we added that, as I'll show you in the tables that summarize the colocalization result. Some of the significant colocalization results, even though the posterior probability is very high, such as this example, may be false positives. And so we added an additional step of filtering to the results to inspect whether the significant colocalizing variants. So for example, in this case, it is a significant EQTL, but looking at this locus compare plot, its GWAS p-value is non-significant. It's above 0.05. So we filter out such cases where the colocalizing variant is not significant for either the GWAS or the EQTL. So in addition to the colocalization analysis to increase confidence that the E or SQTLs are may be the underlying causal mechanism for a GWAS locus, we then applied two-sample Mendelian randomization to the co-localizing QTLs and GWAS loci, both for POAG and IOP. So this is another approach that many people in the field have been applying to assess causal relationship between molecular QTLs and GWAS loci. It's important that the population background of the GWAS and of the QTL match. And so we applied as a primary test, the Wald ratio that looks at the ratio of the beta coefficient from the complex trait GWAS to the beta coefficient of the EQTL or SQTL, and use an inverse variance weighted method when there were multiple independent EOS variants that we use as instrumental variables for the MR analysis. We applied also several other tests as a sensitivity analysis, and the eager intercept test and MR Presso global heterogeneity test to test for horizontal pleiotropy tests. And this analysis was really mastered by Skanda Rajas Sundaram. It was a clinical research fellow visiting Mass Eye from Imperial College London. And he, together with Pooja Mehta from my lab, applied this to all of the thousands of co-localizing EQTLs and SQTLs in the POAG and IOP loci, the various QTL type by tissue, by GWAS loci combinations. And we found significant supportive evidence for causal relationship for about 3/4 of the genes at a Benjamini-Hochberg FDR below 0.05 that were robust to the influence of horizontal pleiotropy. And identifying both, suggesting both E genes and S genes that are potentially affect both POAG and IOP, such as TMCO1, which is the second strongest association with glaucoma or GAS7, and then some genes, DGKG and PC2, that the retina EQTL co-localize with the glaucoma and IOP loci, and also identify genes with support for association with IOP, but not POAG, such as an HLA-B gene or another transporter, and genes that had significant MR associations with glaucoma, but not IOP, suggesting IOP-independent mechanisms that may be important for affecting the sensitivity of the retina ganglial cells, such as the CDKN2B antisense, the RARE and YAP1 genes. So the tables that summarize both the colocalization and the MR results from these analyses, they're supplementary tables in a publication that came out earlier this year, but they're also now, thanks to Maria and the whole team, are now on the Vision Genomics portal. And I can maybe just pause here very quickly, just go to the portal and show you. So do you see my screen?
Yes.
Okay, so you see the portal.
Yep.
So we have if you go to the colocalization summary, we have a summary table. Each row pertains to a given POAG or IOP locus. And so you can, for example, select only one of the traits. So for example, the POAG cross ancestry trait. And then you can also subset on a gene. So we'll look at GAS7, for example. And here we have a lot of information on, how many genes were tested. So how many E genes and S genes are tested here? 30 genes were tested. Many more EQTLs and SQTLs were tested because there may be more than one tissue in which this EOSQTL is significant. So for example, in this case, the GAS7 EQTL co-localized both in fibroblast and an artery tibial. We have information on the direction of effect. So for example, here it summarizes, in this case, the EQTL, the E is for EQTL, S for SQTL. It summarizes and tells you that in two tissues that co-localized, they both have increase of expression of GAS7 has an increased effect on POAG risk. And we report the max posterior probability clip from eCaviar or RCP from NLOC. And so in this example, GAS7, it both co-localized based on eCaviar and based on NLOC. And then we have some additional information on the summary statistics of the GWAS locus. To get more information at the variant by the GWAS locus by E or SQTL variant, by gene, by QTL type, by tissue, you can go to the eCaviar or NLOG colocalization tables, and just takes 1 second to load. And if we were to look here at GAS7, for example, then it gives you information at the individual variant level. So here the variant chromosome 17 position, the ref alt. If we expand here, we can see the interval size that was used for the colocalization analysis, how many variants were tested, the summary statistics of the QTL and of the GWAS, the posterior probability, the probability that the variant is in the causal set, and then the relative direction of effect of the QTL on the GWAS. In this case, it's the same relative, and this is the posterior probability. I see there's a question.
Yeah. Ayellet, there's a question in the chat. It's on MR. It says for the MR analysis, it might be a little late, but we'll do it now. Do you require a minimal number of instrument variables at certain p-value threshold is the question.
Yeah, that's a good question. So we do LD pruning. We require that in this analysis, we required that the EOS QTL p-value would be below 510 to the -6, if I'm not mistaken. And in some cases, FDR below 0.05. We required, we present in this table results that had also were based on one instrumental variable, but we do note in our paper that 3 and more provide more confidence in the results. And the horizontal pleiotropy tests can only be applied if you have 3 or more instrumental variables. So I hope that answers your question. And that's a good segue into looking at the Mendelian randomization results. So as you can see, this is the number of variants that were tested as instrumental variables. This is the variant filter, so the p-value below 510 to the -6. And we do have, as you saw, we do have genes with 1 instrumental variable that were tested. But those should be, considered with more caution. I see there's another question.
Yeah, one's from Laura. Laura is developing colocus. I'm glad she's on the call. Her question is, what is the criteria for R squared and or D prime between independent variables?
Yeah, that's another great question. So we, if I'm not mistaken, we use an R square above 0.1 to prune out dependent variables. But I can go check the method section. But the LD pruning we did was, if I'm not mistaken, an R square of 0.1. Okay, great, thank you. And so in the Mendelian randomization table, we have information on, this is, for example, a HLA-C gene in the European POEG. So we only tested the European, we only applied Mendelian randomization to the European subset of the POEG meta-analysis, and the IOP is primarily European, just so that we don't have confounding from different ancestral backgrounds. So in some cases, we note that we might be less powered compared to the colocalization that was applied to the cross-ancestry meta-analyses. And so here you have the results from the Wald ratio test or the IVW test. Yeah, the p-value and the FDR corrected q-value, and then results from the various other tests for sensitivity analyses, and the test we ran to test for horizontal pleiotropy. And then we note whether it passes or not the pleiotropic test based on a 0.05 significance cutoff. We also note here if it falls in a POEG cross-ancestry locus or in a POEG European or IOP locus. And in this example, it falls only in the POEG cross-ancestry locus, and it colocalizes with a POEG cross-ancestry locus based on eCaviar, but not with the European or IOP GWAS loci. So if there aren't any other questions about these tables, I'm kind of reviewing them quickly just so that I want to cover a few more points. So I'm kind of running out of time. There's a description of all of this in our paper, and then this pipeline of the various tests we ran. We've Pujamed in my lab has implemented this on GitHub, and kind of all the code originally from Scanda. Thanks to Scanda. So just very briefly, we then asked whether these colocalizing genes based on EQTLs and SQTLs with the POG and IOP loci, if they showed enrichment in specific ocular cell types. And for that, we analyzed single nuc RNA-seq data from cell atlases that Josh Sain's lab built, both of 6 tissues in the front of the eye, which include the cornea, limbus, and the trabecular meshwork Schlemm canals. That's the conventional outflow pathway, and the ciliary muscle. That's the unconventional outflow pathway, iris and lens, and then also tissues in the back of the eye, in particular, the optic nerve head, the optic nerve, the surrounding peripapillary sclera and choroid, and single nuc RNA-seq from retina, both peripheral and macular from Ray Chen's lab in Baylor College of Medicine. And we applied a method called Eclipse that we previously developed. It stands for enrichment of causal loci and identification of pathogenic cells in single cell expression regulation data, that is based on mapping genes to GWAS loci based on EQTLs and SQTLs, but also adding in additional mapping from open target genetics using other functional genomic data such as PQTLs, high-C data, deleterious protein coding variants. We then compute a cell type specificity score per GWAS locus and tissue based on the cell type specificity of the genes that map to the locus. And then using a Bayesian Fisher's exact test, we assess the enrichment of cell type specific loci amongst a set of GWAS loci for a given complex disease or trait compared to a null set of GWAS loci that are associated with traits that are not related to the tissue of interest. And then cell types that show significant enrichment and an FDR below 0.1 or 0.05 are proposed to be causal to the disease or trait, and genes that map to the GWAS loci that are above the enrichment cutoff are proposed to affect the disease through that cell type. And you can read more about this on in a publication, Erasla et al., and also in a preprint, and we have the code available on GitHub. So I see I'm running out of time, so I'll just kind of go briefly through some of the highlights of the results. We found that both genes that map to glaucoma and IOP loci were significantly enriched in fibroblast cells, both in the trabecular meshwork and the ciliary muscle. And so the color is proportional with the fold enrichment and the size with the significance. And we found that the genes associated with that colocalize with IOP were significantly enriched in parasites, as well as in lymphatic and endothelial cells and Schlemm's canal cells. And so this part suggests that the unconventional pathway, the ciliary muscle, plays an important role in regulating IOP, in addition to the known important role of the conventional pathway in regulating not only IOP, but also POAG risk. Looking at the retina, we found significant enrichment in Mueller glia cells and in astrocyte cells for the POAG loci. When we remove those that are also associated with IOP, the enrichment remains for POAG, but not for IOP. And interestingly, we did not see any enrichment in the retina ganglial cells themselves, suggesting that the macroglial cells may play an important role in supporting the sensitivity of the retina ganglial cells, and important to consider in drug and gene and cell therapy design. And the astrocytes and Mueller glia cells have been shown to be reactive at early stages of glaucomatous conditions when the RGCs are still intact from various studies in mice and others. Looking at the optic nerve head and posterior tissues, we saw enrichment of the POAG-only genes in astrocytes and oligodendrocytes, the IOP-only genes in endothelial cells, and genes that are associated both with POAG and IOP in fibroblasts, in Schwann cells, oligodendrocyte progenitor cells. And we can look at the distribution of these cells in the various tissues, finding that the fibroblasts were primarily in the peripapillary sclera, the region that supports the optic nerve fibers, and the astrocytes were primarily in the optic nerve head and as well as in the optic nerve. And there's another class of fibroblasts that were enriched in the choroid, as well as also in the optic nerve head. And we can then look also at the genes that are proposed to affect glaucoma through dysregulation in a particular cell type in the eye tissue. So for example, this is looking at the cell type specificity of the top ranked genes and astrocytes in the optic nerve head that co-localize with glaucoma. And so, for example, a retina-specific EQTL acting on DGKG, deacylglycerol kinase gamma, showed the strongest astrocyte-specific expression and is proposed to affect glaucoma potentially through this mechanism. So just given time, I just want to kind of talk about the future directions and the implementation of kind of being able to visualize a lot of these data and data to come in the Vision Genomics portal. So what about the 40% of GWAS loci for which we did not find colocalizing EQTLs and SQTLs? So we're in the process of building a GTEx resource for glaucoma relevant healthy eye tissues, both the trabecular meshwork Schlemm's canal, the ciliary body muscle, the macular and the optic nerve head. So we've collected 10 different eye tissues from 100 postmortem donors. And in collaboration with the Broad, we've performed whole genome sequence on the first 50 donors and now just sending the second batch of 50 donors to the Broad. So we'll have whole genome sequence on all of the donors, RNA-seq, bulk RNA-seq on both the outflow pathways, the optic nerve and macula. And in collaboration with Tim Cherry from Seattle's Children's Hospital, we're performing a TAC-seq on 4 of these tissues from 3 donors. And so we'll have EQTLs, SQTLs, and open chromatin regions on these tissues. And just as a first kind of quick look into the gene expression of genes that fall in POAG loci where we did not find any colocalization results, we see that they have specific expression, different classes of groups of genes in these various eye tissues. So the pilot ENSQTL analysis in these eye tissues is coming soon, and we'll be excited to investigate the glaucoma and IOP loci with these eye relevant QTLs. And so the Vision Genomics portal, we're very excited to have partnered with Noel Bird and Jason Flanick and their team, and particularly with Maria and DK and Trang, and to be able to build a portal we can visualize and inspect GWAS associations with glaucoma, but also other ocular traits in relation to QTLs and open chromatin regions and gene expression from eye tissues and cell types, but also from other tissues such as GTEx. And so this is just really a pilot that was just put up to production, as Noel said, just very recently. So to start with, we have a visualization, a locus zoom visualization of a GWAS POAG locus here. This is the SLC2A12 locus, where the color coding is relative to LD relative to the lead GWAS variant. And we began by visualizing the EQTLs, adding tracks for the EQTLs, and we plan to add soon tracks for the SQTLs. And so this is an example. I just realized I'm not on slideshow mode. So I'll just go back a second and see a bit better. There we go. So we can both group the results by tissues or by genes. So here we're showing each track as a tissue. And if we hover over the SLC2A12, it highlights the variants that are significant EQTLs for SLs for this transporter. In first track is in brain, then in nerve, and then in retina. And so we can see here that there's an EQTL in the region of the lead POAG GWAS variant both in brain and retina, and a separate one in nerve tibial. And so these are the 2 genes that co-localize with this locus. If we hover over the TBPL1, this is the only EQTL shown here in red in nerve tubule that's significant for this gene, suggesting that providing more support that it might be the EQTL and the SLC2A12 that might be relevant. So we're working with the team also to add the colocalization and MR results to these tracks. And here's grouping by gene. So each row is a gene, and the color coding is for the different tissues. And there's a drop-down box where you can choose which tissues to look at. So I only subset it on a couple, but there are actually a few more tissues that have significant EQTLs for these genes. And so, for example, when I hover over retina or brain, that highlights this region, or if I hover over nerve, that highlights a separate locus, a separate EQTL for these genes that is further away from the GWAS locus. And so we're working on adding additional information on the effect size and direction of effect, either based on color or arrow, and adding either subsetting on variants that pass the colocalization and MR significance, or adding different colors to specify those variants. And features that we're kind of interested to add to the portal, and interested to hear if there are other suggestions from the community. Initially, we'd like to add locus zoom plot of the EQTL and SQTL below that locus zoom of the GWAS, so we can more closely see how they align, and to the side, a locus compare plot of the GWAS by QTL, similar to what Laura Scott and team is working on. We'd like to add a track of the SQTL, so gene by splice event by tissue, and an interactive splicing gene model that connects the various splice events to the SQTL, the variants that are affecting the various splice events, with the direction of effect and how it relates to the GWAS variant. And as I mentioned, adding information on the effect and direction, adding on the colocalization MR, and also adding tracks or violent plots of gene expression in eye and GTEx tissues, both at the tissue and single cell level. And then also, I haven't shown you, but below the EQTL, we're working on adding tracks of additional epigenetic data, such as the ATAC-seq data that we're building for the various glaucoma relevant tissues. And here's kind of summarizing the various data sets that we're planning on adding. So we're also planning on adding, starting with existing data, we'll be adding both bulk and single cell ATAC-seq in the developing and adult retina from our collaborator Tim Cherry from Seattle Children's Hospital, and eventually also other epigenetic data such as cis-regulatory elements, super enhancers, and Hi-C in retina from Anand Swaroop's lab. Other molecular QTLs, we recently published a paper with Anand Swaroop on detecting DNA methylation QTLs in retina, so we're excited eventually to add those as well, and to add bulk and single cell expression data from the bulk expression data from the outflow pathway optic nerve head and macula, and single cell expression from available human cell atlases, such as from Josh Sains and Ray Chen's lab. And then, of course, we welcome other functional genomic data from eye tissues from the community. So with that, I'll just end. Oh, yeah, and as I mentioned, the initial focus is on glaucoma, but we will be adding additional GWAS of additional ocular diseases, including AMD, myopia, diabetic retinopathy, and imaging features. And as they're probably aware, there's also recently an ocular knowledge portal was launched, and so we're interested to kind of have some interaction with that portal at some point as well, and then also have our features eventually adapted to knowledge portals of other diseases. With that, I just want to end by thanking the members of my lab that did a lot of this work, in particular Andrew Hamill, Pooja Mehta, Leo, John Rohan, and Jali Wang, Sudeep Mehrotra from our Bioinformatics Center at Mass Eye and Ear, and our various collaborators, GTEx and Anand Swaroop's lab, Tim Cherry, the single cell projects from Josh Sains and Ray Chen, and the GWAS Consortium for the Glaucoma Genetics Consortium, led by Janie Wiggs and David Mackey, and Anthony Kawadja led the UK Biobank IOP GWAS. And then, of course, it's been wonderful, and we look forward to developing a lot of these features with Noel and Jason's team. So I'll stop here, and I see there's a question.
Yeah, I think that's like a jointly answered question, Ayellet. That was awesome, Ayellet. And I love hearing about your work, especially like knowing you as long as I have. It's just really cool, just from a personal perspective too. So that's awesome.
Thank you. Thank you.
We have one question that's in the chat is for Bior. Do you share similar pipelines starting from cis-eQTLs, cis-SQTLs to Colo/MR to ensure consistency in data analysis? I think that's a question for both of us. Ayellet, you make all of these accessible through GitHub. Is that fair to say?
Yes, so we've made a lot of our pipelines or tools available through GitHub. The EQTL SQTL pipeline, we've been using the GTEx pipeline, and I think as now we're computing the EQTLs SQTLs, we're kind of saving our pipeline, because as with any project, you might make some modifications or edits. So we'll make that available on GitHub once we publish the work.
And I think in terms of Bior, so what we'll do is we'll make those GitHub repos accessible through Vision Genomics so people can link to them rather readily. But then more broadly, I think, so your question broadly is, our research platform at large is actually aiming to do this more readily for all types of genetic and genomic data, where a user can load their own data and run a suite of tools that we have. We haven't done Suzy or Colocas yet there. That's coming, as you heard in the beginning of the talk. But right now you could do meta-analysis, you could do MAGMA, you can do LD score regression, and you can do that on your own data and publish a Bior portal that is sort of private, and then you can make it accessible. But you probably want to work with us on that, because we are sort of building this out. So please contact us. We're happy to work with us. That's sort of part of what we love to do, as you can see from the work that we've been excited to do here. But try it yourself if you want to, but it does require a little bit of piecing things together, because we do like to customize this for people as they go. But there's some basic stuff you can do without even our help at all. Hopefully that answers your question, but feel free to email me afterwards, and we can definitely set up a time to talk directly about it. Other questions? Fabulous. Ayellet, I want to thank you and your team. Always lovely to work with you. I'm so glad, and I love this portal. I love what its mission, and I think hopefully you will use it, give us feedback so we can make it better too. Mackenzie, when is our next webinar and focus group?
So our next is going to be in 1 month, and so that date is going to be, so it'll be a focus group, and it will be on May 9th, sorry.
And I think, if I recollect, our ocular disease group is coming too. So this will help us convert on the eye. So as I've learned about the eye the eye is fascinating from a biological standpoint, also in terms of like the way you address the scientific questions. So that's why these community portals really do feed into that concept of how you approach scientific questions, and then eventually they can coalesce together. So look forward to seeing everyone there at that session, and we'll advertise that and share that in our various social media channels. Thanks, everyone.
Thank
Appreciate
you.
it. Thank
Thank you for having me.
All right, always.
Bye.
Take care. Bye-bye.
Bye.
Bye.